Good evening, everyone. Welcome to Botany Insider's free revision course. So, we are starting off with a free revision course on our YouTube channel for exactly 1 month. Today is 27th of November. So, we'll be continuing with our revision classes till the examination, right? So, examination dates are between 26 to 28. So, we'll be getting the revision done of the various topics, of the various important topics for exactly 1 month. So let's wait for students to join in, then we'll begin with our class now, right? We'll be learning about, today we'll be like targeting out the biochemistry, and then we'll be proceeding ahead with other units as well. Let's wait for a minute and then we'll start. Please let me know in the chat section whether the audio and the video is clear to you or not. Okay, thank you, Khushi. Let's begin with the class now. Right, so let's talk about the biochemistry unit. First thing first, it is one of the most scoring unit for our upcoming examination, right? It's one of the most scoring units. If you know about the topics, if you have like prepared the topics well, you can easily target out the questions from here. First thing, in addition to the same, in this entire series, we'll be dealing with important topics list also, so that it can help you out in understanding that whether in the last 1 month are you actually like focusing upon the important topics or not, right? Let's begin. Before starting, we are or there's a course going on for the June 2024 students. If you're targeting out the next examination, then we have a full course ongoing for the June 2024. The link for each and every course, all the things is in the description box. If you wish to enroll, you can download our application and get yourself enrolled. In addition, for the upcoming December 2023, we have a mock test series going on in which you get 100 plus mock tests. So, those of you who are willing to get this, you can get the entire 100 plus test mock test series from our application itself, available in very affordable prices, right? 599 is the price for the same. Okay, let's begin. So this is the entire syllabus of your upcoming biochemistry unit. We can say you have structure of atom, molecules and the chemical bonds. Then we have composition, structure and function of the biomolecules. Then we have a number of stabilizing interaction, that is a number of bonds that we have. Then we have the principle of biophysical chemistry. Then you have the concept of your respiration. Then you have principles of catalysis, that is the entire topic of enzymes, conformation of proteins, in which we learn about Ramachandran plot, secondary structure of protein, domains, motifs, etc. Then you have the conformation of nucleic acid, one of the topic from unit number 3, right? Then we have stability of protein and nucleic acid, and finally the metabolism. This is about the course. This is about the entire listing of the content. Now talking about which is the scoring part, from which topic you can easily get question. If you analyze the very recent papers, very first thing, you can easily get a question from chemical bonds. This year also there was a question asked from chemical bonds. You can easily get a question from the enzyme topic. Every year we can expect at least one topic from enzymes. Then you can expect a topic from nucleic acids conformation, majorly in match the following type, right? Then in the stability of proteins, in recent year there was a question asked based upon the digestion of protein, the protein digestion, various enzymes used, various enzymes which work in various different areas. So you can work on, or you can expect one question from this area as well. In 23, there was a question asked based upon carboxypeptidases, right? In the recent last year, you got a question. Before that, we didn't see a question from carboxypeptidase. So it was a new topic that was taken up. So you should remember that. Carboxypeptidase is what? It's an exopeptidase which works on the C terminal of your amino acids, or on the C terminal of our polypeptide chain, right? There's a complete listing for the same. You have three major classes of carboxypeptidase, which is A, B, C, right? Remember all these things. Then from Ramachandran plot in 2022, there was a question asked. So you can expect a question in the upcoming examination as well. Then from your vitamins as well, because from last 4-5 years, we have not received a question from vitamins. So this time they might surprise you with either a match the following or either a metabolism question from vitamins. So work upon all the topics very effectively, right? If and in addition to that, there was a question asked from hemoglobin degradation in 2022 also and in 2023 as well. So from last 2 years, we are getting question from hemoglobin as well. So do not skip the degradation part of hemoglobin. Very, very important, right? So, see, these are the most important topics. First one is your amino acids. Then, from the basics of protein, you can get a question. From structure of protein, again, you can get a question. Then you have from hemoglobin, very easily question can be there. From enzymes regulation part, that is when we learn about the enzyme kinetics, you can get a question from here as well. Vitamins, easily you can add on protein digestion portion as well. You can add on protein sequencing question as well, right? So these are the most repeated and the most important topics for your from unit number 1, specifically from the in the upcoming examination, right? So let's discuss about few of the topics so that you can get an idea for the same. For example, what are the various types of chemical bonds? So we majorly have four chemical bonds. You have ionic, or on a broader level, you have two major classes of chemical bonds based upon whether there is sharing of electron or do we have entire transfer of electron, right? In the sharing of electron, you have the formation of the covalent bonds. In the transfer of electron, you have the formation of non-covalent bonds, right? Covalent bonds are more stronger in nature. Why they are stronger? Because the electron is getting shared with the other one. So there's a higher tendency for the two atoms to stay in close proximity, because both of why do we have a bond, like why do two elements come in contact of, or why do they interact with the bond formation, so that they can increase their stability. And when the stability of both of the elements is involved, they'll be in much more closer association, they'll be in a much more stronger association. So sharing leads to the formation of covalent bonds. Covalent bonds are stronger, reason being they require higher energy for breakdown. See over here, when the two non-metals share the electrons, for example, oxygen atom, you have these oxygen atoms, then they both will be sharing two electrons together to form up a oxygen molecule. Then in water as well. Then you have the non-covalent bonds. So non-covalent bonds are what? In which the electron is completely transferred to the other element. In this also there is like energy required to break them, but comparatively very less energy is required for the second segment, for the non-covalent bonds, which includes your ionic bonds, which includes hydrogen bonds, which includes metallic bonds. Ionic bonds are what? Between, for example, NaCl. So Na plus, Cl minus. One of them loses one, other one is gaining one electron. As a result, there's a stability. So the one which loses the electron or which donates it, it is known as cation, which will be getting a positive charge. The other one is an anion, which gets a negative charge. Then you have hydrogen bonds. So when a hydrogen atom electronegative, like it attracts a electronegative atom towards itself, so it remains in close proximity. For example, in water we see hydrogen bonds getting formed in. Then you have metallic bonds as well. So positive metal ions when attract or when they conduct the electrons, when they attract the conducting electrons, we call them, or this is what a conducting, or this is what a metallic bond is all about. Very important, very easy. You can easily like revise everything from this segment. Then you have, see, there was a question asked in 2019 where you have various bonds given and you have their energies for the same. First one is hydrogen bonds, then you have van der Waals forces, covalent bonds, and ionic bonds. And over here you have their energies related to the same. So van der Waals forces, covalent bonds, ionic and hydrogen. So van der Waals forces should be the least energy forces. So the least out of the four values given to us is zero point 0.5. So Q should be marked with your first statement. If Q getting marked with the first statement, I have either first or third as the correct answer, the second and the fourth as the correct answer. I can eliminate first and third with just one option. Then you have covalent bonds. So covalent bonds are of highest energy. That means are getting marked with your third. That is your uh, covalent bonds with the highest energy. Then next is between your hydrogen and your ionic bonds. Ionic bonds are comparatively stronger than your hydrogen bonds. So over here you have 40. So S getting marked with the second one and hydrogen bonds getting marked with 3 kCl. So I can easily get my correct answer as option number D. Right? You should be very much thorough with solving out the questions. You should be very thorough with learning how to minimize your time and maximize the best output possible in the examination, right? I hope the question is clear to all, right? Then there are some amino acids with some unique properties. For example, side chain with unique property. First is glycine. Why glycine is unique? Reason being the side group and your other group is, both of them are H, so it's the simplest one. Then cysteine, why is it unique? Because it can easily make up a sulfur bond. It can easily make up a disulfide bond with the other cysteine. Again, important, exceptional. Only cysteine is able for, or only cysteine can actually form a disulfide bond with the amino acid. No other amino acid can form a disulfide bond. Reason being, cysteine have a free SH group. Only cysteine has a free SH group. Now, you can easily get a question over where they can ask you that why methionine does not form a sulfur bond. First thing, cysteine and methionine, both of them have sulfur in them. Both of them have sulfur atom in there, but only cysteine can form the bond because methionine does not have a free sulfur. There's a lot of steric hindrance in its structure, and as a result, you can see only cysteine forming the bond. Then you have proline. Proline is again unique. Why is it unique? Because of this different chain in it, right? Because it is complete, it can form kinks in the structure of your secondary structure, so it can easily form in the bond with it, right? So remember about all these things, very very important. Just check whether it's clear or not. Yes, Geeta, you can join in the course with installment option, no worries in that. You can use the installment and join in our full course. It's ongoing right now. You can download our application. I'll guide you. There's a additional 400 off coupon going on available till today. So if you wish to enroll, you can get enrolled in the same accordingly. Okay. Everyone just check whether everything is clear or not. We have various students who join via installment options. So no worries in that. You can join in very easily. Okay. Okay, Harshit, you can help your classmate also for the same. Please send the link. She'll get help for the same. Okay. Right. So I hope till here things are clear. So unique properties, which are the unique properties carrying amino acid? First one is glycine. Second is cysteine. Third is proline. Why cysteine is unique? Because it's having a free SH group. Why proline is unique? Because it can disrupt your secondary structures. Okay. It can disrupt your secondary structure. So remember, though side chain have hydrophobic character, it has a unique property of creating kinks in the polypeptide chain and disrupting the ordered structured sequence. Very, very important because this is one of the exceptional case which you can keep a note of, because in Ramachandran plots question, every time you have to remember two segments, that glycine is found in the maximum region and proline is not found in the Ramachandran plot. Okay, glycine is found in the maximum region and Ramachandran plot is not found in the entire plot, right? I hope it's clear. Right, let's move forward now. Next, let's talk about the polypeptide chain. Let's talk about what a polypeptide, or how does a polypeptide chain gets formed. So you have the first amino acid, you have the second amino acid. When you look into the structure of the amino acid, you have a amino group, you have central carbon, you have carboxyl group, you have R, and you have H. Okay, this is the variable group. Now R, and now on the second amino acid also same thing will be there. So from the first side, the C terminal will be participating. From the second side, your N terminal will be participating. With the participation of these two groups, you will be having what? You will be having the elimination of one of the water molecule. And in addition, one ATP will be utilized in this particular process. As and when one of the ATP gets utilized in the process and you have elimination of one of the water molecule, what do we have? We have the formation, or you have the like water gets eliminated and a bond gets formed between the two. What is this bond known as? This bond is known as your peptide bond, right? You can easily get a question from this segment because CSIR nowadays is focusing a lot on the basics, right? It is focusing a lot on the basics. So you can easily work upon the same. Remember about this, that there is expulsion of one water molecule in the entire segment, right? I hope it's clear. Next, we have, like, what you should revise is the hemoglobin degradation. Okay, Geeta, you can use this particular link for the course, right? So, talking about the next thing, that is about the hemoglobin degradation. From this, also, there was a question asked in the recent paper. So, we have the aged erythrocytes. They enter, or they are taken up by the macrophages. Macrophages, from there, the globin part and the heme part get separated. The heme part is further like degraded. From the globin part, we have the amino acids taken up and they are either reutilized or degraded. Then the heme part, with the help of heme oxygenase, Fe3 plus and carbon monoxide getting released off, you have biliverdin getting formed. First thing, biliverdin gets converted into bilirubin with the help of biliverdin reductase. Then bilirubin is released in the blood where a complex is formed known as bilirubin albumin complex. Then from there you have this bilirubin albumin complex entering into what? It is entering into your liver. So in the liver, you'll be having bilirubin converting into, with the help of bilirubin gluconeuride transferase, converting into your bilirubin diglucuronide. Then you have in the intestine or in the kidney, finally, this bilirubinogen, if it is in the kidney, then it will be converting into urobilin and finally released from the body in the urea or in the urine. And then bilirubinogen, if worked upon microbial enzymes, then it will be getting converted into stercobilin and then passed out from the body in the fecus. Okay, so remember about this. In the same exact thing, from this particular step and from the first thing formed as biliverdin and not bilirubin, you can easily, like from this step, the question was asked in the examination. So keep a note of this, that first we have biliverdin getting formed and then bilirubin, and also there is expulsion or there is release of CO, that is carbon monoxide. Okay, in the question, four options were there. First was related to which of them, like either whether it is biliverdin or bilirubin, which one is getting formed first, and then you have whether it's the heme part or is it whether it's the carbon monoxide or the nitric oxide, which one is released. So you have carbon monoxide being released and biliverdin getting released or getting formed first. So remember about this very, very important concept. And if you wish, you can draw this entire thing or take a screenshot, whatever is helpful for you. Take a screenshot and remember the entire pathway for the hemoglobin degradation. And remember this. I've given you a very good revision for the same. So it will help you out in the upcoming examination, right? Next, we have one more question. So this question is that in an alpha helical polypeptide, the backbone hydrogen bonds are between which of the following? When we look into the structure of protein, so you have the primary structure, then the secondary structure, then the tertiary one, and then the quaternary structure. So four of the structures levels are there. In the primary, you have the linear chain. In secondary, you have alpha chain or beta alpha helix and beta sheets, then tertiary, then quaternary. Then in the alpha helix, if we look in the alpha helix, we have hydrogen bonds getting formed in between the consecutive amino acids that we have. So from the similar concept, the question has been asked. I have explained this thing. There's no other book required. You can just remember this. That's more than enough for you to get through with this entire hemoglobin degradation. Just remember this, and you need not to go into major depth after this segment. This is more than enough for you. And if you want to refer, you can use CART cell biology. In that, you can easily find a book. You can easily find a hemoglobin degradation concept. Okay. So over here you have hydrogen bonds. So in the alpha helical structure, hydrogen bond is formed between the first and the fifth one. Between the first of your, what do you say, amino acid and then the fifth one. So there's a hydrogen bond getting formed between first and fifth, second and sixth, so on and so forth. So if you go into the theoretical concept for the same, answer over here should be option number C, that is CO of N and NH of N plus 4. This is what you have to keep in mind, that there's a, it's not blur, just take a screenshot or WhatsApp me, I'll share it with you. It's very much readable. Wait, let me shorten it, it will be more beneficial. See, it's more than enough for you to do it. Like there's no other thing required. It's enough for you. If you go ahead with this, you'll be easily able to mark the answer for this question, right? For this particular question, the answer is CO of N and NH of N plus 4, right? So this is about the revision of all these things. Now tomorrow we'll learn how to solve out this question. This is again a recent year paper, that is June 2023 evening shift paper part C question. We'll be understanding how to solve it. It's a very, very simple question. You just have to remember one important formula and you'll be easily able to solve such a question. Bilirubin's original color, like what I have learned, it is red orange, like reddish orange color. I have, wherever I have read, or according to my knowledge, it is reddish orange in color. But based upon various different sources, yes, they talk about different colors. I also have like came across this particular anomaly, but to my knowledge, it is red orange in color. Wherever I have read, it is majorly red orange written down. Okay. So you can keep a note accordingly, right? So I hope this particular session helped you out. In addition, we'll be discussing the further segments tomorrow. We have a mock test series going on for the December 2023 students. 100 plus tests are there. The price is only 599. If any one of you who wish to enroll, you can download our application and get yourself enrolled in the same. In addition, there's a full course going on for June 2024 students. Those of you who wish to enroll in this course, if you are like targeting out the June 2024, you can get yourself enrolled in this. It is available in very affordable prices. The complete 6-7 month courses till your main examination prices 4949 only, with hard copy notes. We'll be delivering hard copy notes to your doorstep. So in addition, all the units including general aptitude, all recordings, all class PDFs available, daily practice question. We are practicing out the questions daily. These are detailed proper lectures, PYQ sessions. Every Sunday, a test is held, regular doubt sessions are held. So, we have tried to incorporate every single thing, whatever you require for your CSIR preparation in one course itself, and that too in a very, very affordable price. It is 4949 only. Installment options also available, as one of the student was asking. I have pinned the chat also for your course. You can directly tap on the link for getting into the course, right? And in addition, there's additional 400 off available till today. We have already a number of students enrolled. So those of you who wish to enroll in the same, you can download our application. Link is in the description box. You can WhatsApp us on this particular number for any query, for any discussion or any type of query that you have related to the course, right? So there are students in the class right now. If you wish to ask anything related to the course or any topic you want me to take up specifically for this entire 1 month, you can ask. Otherwise, we're done with the class, right? Tomorrow also, I'll take up enzymes topic, few different conceptual topics. Join in the classes every single day. We'll revise and we get done with the topics in the best of the ability, right? So, all the best for the upcoming examination. Good luck. Take care, everyone. Let's meet tomorrow now. Bye, everyone. Take care.